This video is part 2 of the nervous system basics, and it's going to be starting in the notes at the place where it says multipolar neurons. This table 11.1, my note is that it's on page 396 in your textbook. What you need to know about the difference between multipolar, bipolar, and unipolar is predominantly that the number of things connected to the cell body is what determines which of these it's going to be. So multipolar means that there's a bunch of branches coming off. Bipolar means there's 2. Unipolar means there's 1. So anatomy is pretty simple when it comes to these. The other thing that you should know is that multipolar neurons are the most abundant type of neuron. You can find them pretty much anywhere, including motor neurons that we talked about briefly back in muscles are multipolar. Bipolar are relatively rare. You can find those in some of your special sensory organs, like the retina in the eye has some bipolar neurons. And then unipolar, you can find those in like the dorsal root ganglia, and they tend to have a sensory function. In terms of the 2 different types of neurons that you have next, it's really just a directional thing. Sensory neurons are taking information towards the central nervous system. Motor neurons are taking information from the central nervous system. So you can even tie those back to some of our structural and functional divisions that we had earlier. Your sensory neurons are going to be part of the sensory division of the nervous system. Your motor neurons are going to be part of your motor division of the nervous system. Next up, you have some definitions that just kind of go along with electricity to get you some stuff there, starting with voltage. Your definition, the top sentence right there, gets you what you need. I do also want you to know that when we're talking about AMP, the unit is going to be in millivolts. We're talking about a minute amount of voltage that can be measured across that membrane. Current. It's the flow of electricity from point A to point B. Current is usually measured in amps. Resistance is opposition to flow. If you are a person who just loves math formulas, then the formula for all of those things is E equals I times R. So that's voltage equals current times resistance. And it kind of just tells you how electricity is going to be working within a system. Now, to tie it to an analogy, this can be any electrical wire. This particular stuff right here, the copper wire, that's a conductor. It has a very low resistance, so electricity travels through that really well. This is the insulation around that wire. It has very high resistance or opposition to flow. This could be like your axon. This could be like the myelin sheath. And so these terms totally relate to the nervous system and how it's going to be sending information to and fro. Next up. Here's where we need to tie it back to stuff we learned way back in Chapter 2 and Chapter 3. Your plasma membrane is a semi-permeable membrane. Some things could get through and some things could not. Here we're talking about electricity. Electricity in your body is an ion thing, and so that's where the chemistry part of it is going to come into play. These little yellow balls could represent any ion that you have in your body, and you need to remember phospholipid bilayer does not want to let ions come through it because this is all nonpolar, and ions don't like to go through nonpolar things. So we're going to have a way to get those ions across, and that's where some of these proteins are going to be helping us out. So leakage channels are gates that are just always open, and they tend to help ions come in or out depending on just concentration gradients. Gated channels are channels that you can open or close depending on whether or not you want a particular ion to come through at a time. For example, in this picture, here is this gate in a closed state. When a chemical comes in and it binds to it, it makes this gate open so that now these ions can go from an area of high concentration to an area of low concentration. So the only difference between these two, always open, can open or close depending on needs. Next, there are 3 ways that we're going to talk about where you can open some of those gates. Chemicals can bind to them and open them, and that's what happened in the picture earlier. A chemical came in and it bound to this gate, and as a result, this gate opened. A voltage can make a gate open, so let's say electricity is already running through, that opens more doors. You can also use physical deformation, so like you touch something and that can open a gate. Next up, electrochemical gradient is a combination of 2 things. A chemical concentration gradient, i.e. there's a high concentration of these orange balls on this side, and a low concentration on this side. They would like to diffuse from high to low. And there's an electrical gradient because in your body, these things are ions, which means there's a charge that's associated with them. Let's say that these balls represented sodium ions. There's a bunch of positives on this side, nothing over here. They would like to go over to the opposite side. And so the combination of the electrical charge plus just the fact that it's a chemical makes that an electrochemical gradient. Next. Resting membrane potential, there's a video in your on your book's website that helps explain that, and we talked about it back in Chapter 3. there is a charge across the plasma membrane of most of the cells of your body. It ends up having a net negative charge to the inside of the cell because of some things we're going to talk about here in just a second. So even when a cell is not doing anything in terms of electricity, it has a negative voltage on the inside of the cell. So when a neuron's not communicating, it is in resting membrane potential. Polarized just means that there is a charge across that membrane. And so that's what we've been talking about all along. Our membrane is polarized when it's at rest. Now, what helps us to make that happen predominantly is the sodium-potassium pump. Although it's not the only thing at play, it's a big part of it. We talked about this way back in Chapter 3. What the sodium-potassium pump does is it pumps 3 sodium ions to the outside of the cell for every 2 potassium ions to the inside of the cell. And it cycles that over and over and over again so that you end up with this huge concentration of sodium ions outside relative to inside, and a huge concentration of potassium ions inside relative to outside. That's the concentration gradient, the chemical part to this. Now the other side is we pumped 3 positives outside for every 2 positives inside, so we end up with this slight positive charge to the outside of the cell. So this sodium-potassium pump is polarizing the membrane by giving us that charge difference, and it's setting up that resting membrane potential, which is the polarized membrane. Next, depolarization. Now, when you're at rest, the voltage in a cell, if you have it hooked up to a mini voltmeter, is negative 70 millivolts. Anything that makes that line go up towards more positive is a depolarization event. Now, when we do a full action potential, we're going to actually become positive. But even just a little bit higher than negative 70, that is still a depolarization event. Hyperpolarization is anything that makes the membrane more negative. And so here we've gone down to, let's say, negative 75. That would be hyperpolarization. Next, there are 2 basic types of potentials that can happen across the membrane. First thing I want you to know is where each one happens. Graded potential happens on the dendrite side of a neuron. Action potentials happen on the axon side of a neuron. So location is different. That's first and foremost. Second thing that I want you to know is just the definition. Graded potentials are short-lived. They get weaker also as you get farther away from the point of stimulation. So this last sentence right here hits on what I just said. The current is decremental. That means it gets weaker as you get farther away from where you hit it. Action potentials, on the other hand, they are what's known as all or none. They happen or they don't. They don't get weaker as you travel. They are the same strength the whole entire way down. If you have ever heard of a nerve impulse, it is an action potential. So action potentials and nerve impulses are the exact same thing. So do not decrease in strength. Also, notice it's the principal way neurons send signals. Again, this is a nerve impulse, what happens with an action potential. We're going to go over this table in class, and so if you don't get it, there are some videos on the website to help you out with that if you happen to be an internet student. Let's see, we will also work on saltatory conduction in class, and there's a video for that on the book's website for my internet students. Next thing that you guys have is refractory period. I have a slide for that eventually. Here we go. What's going to be happening during an action potential, it's all about gates and when they open and when they don't. When we do an action potential, sodium gates open. That's what starts the whole process because we open these sodium gates, sodium ions flooded into the cell, bringing with them their positive charge. That's why we become more positive. Here's where the weird thing comes in. During this refractory period, this green that you have going on right here, the sodium gates are already open. Since they're already open, you can't open them again. So there is this period of time where your neuron is said to be insensitive, and that's the refractory period. This does 2 things for us. It makes sure that every action potential is a single, concrete, discrete bit of information, and it makes sure that our impulses always travel one way. We don't get backwards flow down an axon, so the impulse always travels one way because of the refractory period. Now, absolute is you absolutely can't re-stimulate that neuron. Relative refractory period means you could re-stimulate it, but it's going to take a big stimulation to make that happen. And so it's harder to start a neuron when it's in the relative refractory period. Next thing that you guys have in your notes is multiple sclerosis. Highlights. You need to know it's an autoimmune disease. You need to know that it demyelinates the neuron, and you can see a picture. Remember, the myelin sheath is that insulation that you have around it. In this particular case, the immune system comes in and it starts destroying the myelin sheath. That slows down impulse transmission, and it can allow for one neuron to communicate with another one accidentally, or what I'm going to call short-circuiting between the neurons. And so that's where some of the symptoms come in for that. Symptoms are all in this sentence right here, so notice what those are. If you were to see a person with multiple sclerosis in a scanner, you would notice these hardened white lesions right here. They're called scleroses. And so the fact that a person has a bunch of them, that's why it's called multiple sclerosis. It's because they end up with a bunch of these hardened lesions where there used to be a myelin sheath, but there is not anymore. There are a bunch of immunosuppressant drugs that are out on the market. This is an old picture, and probably these guys aren't even used all that much anymore, although I'm not sure. I don't stay up on my multiple sclerosis drugs. But there's a bunch of newer ones that come out on the market. There is not a cure for multiple sclerosis still at this point. Stem cells, once again, seem like they might be helpful for people with MS, but that therapy is currently not available in the US. All right, that's all for this particular one. We'll start with the synapse in the next video.